right, chapter 16.2 in OpenStax talks specifically about chemical defenses, and we've mentioned a few already, but I want to spend a little time now on complement, because that's something that's a lot of people have maybe never heard of before. So it's kind of a strange name, complement. This is a series of proteins that are present in the blood. Maybe about 30 different proteins. And what they do is help other aspects of the immune system. So I guess their action complements lots of other things in the immune system. So maybe that's where the name came from, but kind of a strange name. Still considered part of our innate immunity. So again, more non-specific responses, going to respond the same way every time that pathogen is encountered. These proteins are considered part of a cascade reaction. So that means one protein acts on another protein that then acts on another protein that acts on, so on and so forth, like a rock, that rolls down the hill and eventually hits lots of other rocks and causes a big mountain slide or whatever. So what do these proteins actually do? Three main functions here. They are very important opsonins. And I'm not sure, I don't think I have defined that before, but an opsonin is anything that's going to make phagocytosis more likely to happen. So in other words, they act to coat the outside of invaders, mostly bacteria, so let's just say bacteria. Coat the outside of bacteria so that phagocytes can more easily engulf cells. Okay, so another important opsonin, as we'll see in a little while, are antibodies. But these complement proteins, some of them will bind to the outside of bacterial cells and make phagocytosis more likely to happen. Some complement proteins act to increase inflammation and chemotaxis of cells. Those two things kind of go hand in hand, as we'll see when we talk more about inflammation. So, in other words, attracts other immune cells to the site of infection. And they can actually act to kill certain kinds of pathogens. They can actually lyse some bacterial cells by this MAC, which is an abbreviation for a membrane attack complex. Okay, so they can not just help other aspects of immunity, but actually cause the lysis of certain bacterial cells. All right, so here's your book is really deficient in any kind of meaningful graphic. So I've tried to find some here to show you, but this is just a little bit of an overview. We're going to get a little bit more detail than this. There's several different ways that the pathways that this cascade reaction can get started. So we'll talk about those three in a minute, but one way is if antibody, and I know we haven't talked about antibodies yet, and we haven't talked about what antigens are yet, we'll get to that. When our antibodies that's produced by our B cells actually recognize some maybe surface protein or receptor of a bacterial cell. So, in other words, our antibodies recognize this antigen complex. Usually, again, this is usually going to be a bacteria with potentially be like a membrane virus, but usually bacteria. And this first complement protein is actually a C1 complex. There's actually 3 different parts to that protein, but it binds to this antigen antibody complex, and that causes the splitting of other complement proteins. Some of those bits will form another protein that act on another complement protein. Those bits become important, so on and so forth. We're going to talk about what some of those bits do. Some of the bits all come together to form these membrane attack complexes. All right, so some of these bits become important for the inflammation and the chemotaxis. And some of these bits actually are important opsonins. They are actually, opsonins. They are actually going to kind of come back up here and bind to that bacteria cell so that some like macrophage or neutrophil would be able to, more likely to eat it. All right, so that's just a little overview. This diagram is from our old textbook, and I'm going to kind of write over the side here, especially what I want you to take away from this, but hopefully this helps you visualize it just a little bit better. So again, here's that first complement protein. We call that C1. This is just one of the ways that the whole protein cascade can be activated. It recognizes, here's an antibody, and here's a bacterial cell would have all sorts of different antigens on the surface. Okay, so that antigen antibody complex is recognized by the C1, and then the C1 acts to split the next 2 complement proteins, C2 and C4. And when those proteins split, we label the fragments A and B. So we get C2A, C2B, C4A, C4B. Some of those fragments will come back together. Don't really need to memorize which is which, but the C4B, C2A, that protein complex is so important, we give that another name. That's called C3 convertase. Okay, and that acts to split that complement protein C3 into C3A and C3B. Okay, now we get some of these bits that are acting on their own. So you can see here C3A triggers inflammation. We'll see in a bit one way that can happen. And C3B acts as an opsonin. So again, that is going to come up here. Oops. C3B bind here to that pathogen, and phagocytes will have receptors for that C3B, and again, it's like handles to be able to hold on to that cell. So let's write this up here. So C3A acts to increase inflammation, and we'll talk more all about inflammation soon. C3B acts as an opsonin. So the B, you can think of the B for binds, and the A you can kind of think of as allergy. That might help you remember inflammation. Okay. So back down here at the bottom, some of that C4B, C2A actually will bind with the C3B to split C5. And then once again, the C5A triggers inflammation. So let's write that up here, C5A. So again, the A's allergy or inflammation. And then this B is going to bind as well, but it's going to bind with the C6, C7, C8, and lots of C9s to make that membrane attack complex. Okay, let me write that over here, C5b, along with, we'll say others. Yeah, well, let's get a little bit more specific. Let's say C6, C7, C8, and C9 to make that membrane attack complex. All right. So that membrane attack complex, this is poking a hole actually in the outer membrane of gram negatives. Okay, and it's actually strong enough to poke through that thin layer of peptidoglycan and into the plasma membrane. We think that this primarily happens with gram-negative bacterial cells. Gram-positives have such a thick layer of peptidoglycan that doesn't really affect them so much. So this is membrane attack complexes are primarily acting on gram-negative bacteria. These other bits, the C3A, the C3B, the C5A, those are all acting on gram-positives as well as gram-negatives. All right, so here's a transmission electron micrograph showing a cell that's in some real trouble, because you would think maybe poking a hole here or there in the cell membrane wouldn't be a big deal, but lots of them, lots of complement acting on this cell, and of course that's going to bring about death of the cell, lots of holes poked in it. Okay, so there's actually, I referred to you, there's several different ways that these complement proteins, remember these are proteins that circulate in the blood, they're actually made in the liver, circulate in the blood, and then they can move into the tissues when there's some kind of inflammatory reaction. How is this process activated? We already showed the classical pathway. We call it the classical pathway because that's the first way that we discovered. But that is activated, complement cascade is activated by antibody bound to antigen. Okay, so we showed that already. Okay, C1X as an enzyme. All right, when that C1 complex gets activated. With the alternative pathway, we'll say C3 is split by pathogen. Basically, this is a lot more complicated than what I want to go into, but C3 is constantly being split at a very low level all on its own. And when it splits and it comes in direct contact with a pathogen, then that causes, and there's some other protein factors present that will cause many more C3s to be split. And with lectin activation, C3 is split by microbial sugars. Okay, bound to lectin. Okay, and I explain that, maybe it's a little bit better to see with a picture. So here's the three ways of activation. Classical, again, antibody, antigen activation there. With the alternative pathway, that was the one where we said C3 is constantly being split. If it recognizes certain receptors on a microbe, then that, and there's some other protein factors present that we're not going to worry about, even more C3 gets split. And with that lectin pathway, I explained this a little bit better, but there are sugars often present on pathogens like bacterial cells, like mannose. It's a monosaccharide that is not found on the surface of our cells. So that's recognized by these proteins, lectins, that are made in our liver. And when that recognition happens, then that causes lots of C3 to be split. So it doesn't really matter which of the three ways that complement is activated. It all results in the same complement cascade. Again, once the C3 here is being split, and we get the C5s and the membrane attack complexes. Okay, so no matter how it's activated, we get a very, very similar complement cascade. All right, there's one YouTube video that you might want. This is the best one that I've found. I'm sure there's a lot of good ones out there. This one moves really fast, so might want to watch it a couple times to let it kind of sink in. But it kind of, it's a an animation and just a quick drawing of the complement cascade and what we get out of it. Okay, so remember all these proteins, this activation happens in the blood, but then all these little bits, these C3As, C5As, are going to move out into the tissues and following, wherever the pathogen is. Okay, and I try to be really clear in the learning objectives, what exactly I want you to take away about complement, because I know all the numbers and everything is a little bit much the first time you see it. All right, so while we're talking about chemical defenses, I want to at least introduce a little bit about cytokines. I'm just going to mention most of them, and we're going to talk a little bit more specific about the interferons. But cytokines, you're going to hear that word a lot. They do a lot. In general, they act on many kinds of cells in order to coordinate the whole immune response. Okay, so this is the way that all of our immune cells talk to each other. We used to call cytokines, them interleukins. So we have many, many different interleukins, IL-1, IL-2, I think it goes to like IL-36. Okay, all these numbered interleukins. But then we started to recognize that these signals weren't just happening between white blood cells. They were acting on a lot of different kinds of cells. Sometimes we use a term so much that it's so familiar that it's hard to change the term. So now we just say that interleukins are a kind of cytokine. Okay, so there's lots of them. We're not going to talk specifically about any of them. There's lots and lots of overlapping functions. So in other words, one interleukin might do basically the same thing as another one. So it gets pretty confusing. Chemokines, just if you hear that word, that's a kind of cytokine. It's kind of causes the chemotaxis of certain cells, but again, just recognize that that's a cytokine. Another kind of cytokine is tumor necrosis factor. So TNF, there's several different kinds of tumor necrosis factors. The name came from, we discovered this I think as a protein in mice that when it acted on mice, that it caused tumors to break away and die. So you'd think, oh great, this is a cure for cancer. When we purify this and add it to humans, it causes such an incredible galloping inflammation that it's enough to kill a person. So there are a number of autoimmune diseases where our immune system is acting inappropriately, that the drugs we give are actually tumor necrosis factor blockers. So Humira is one that's advertised a lot on TV. for conditions like rheumatoid arthritis or Crohn's disease, and it's actually blocking the body's production of tumor necrosis factor when that production is inappropriate. I do want to spend a little bit more time talking about interferons. That's a kind of cytokine. And there's a couple of different kinds of interferons. There's interferon alpha and beta. Those are the ones that we're going to spend a little bit more time talking about here because they're very important to curb the spread of viral infections. It's really our innate immunity, really one of its very few answers to a virally infected cell. One of the very few ways that it tries to, our innate immunity is able to curb viral proliferation. There's a different kind of interferon called interferon gamma. It's a totally different function that's part of adaptive immunity, and we're not going to worry about that, okay? So, so before I get to that, I forgot to mention that we can think about how cytokines act. They act in three different ways in our bodies. Autocrine, meaning that some cytokines actually act on the cell that secretes it. So a lot of immune cells, they'll secrete some cytokine and they'll actually have receptors for that cytokine that kind of tell it to do some kind of function. So that's an autocrine function of cytokines. Some cytokines act on nearby cells. Okay, so and here's a nearby cell that has receptors for that cytokine. And some cytokines, the function is considered endocrine, meaning that they travel throughout the whole body and get in the bloodstream and act throughout the body. Okay, so then I just, to finish up today and talking about our chemical defenses, I wanted to show you a little bit more about how those alpha and beta interferons, which are called type 1 interferons, alpha and beta, how primarily the way that they act. So interestingly enough, they don't act on the cell that really needs it the most. So what I mean by that, here we have some cell in our body that has been infected by a virus. So that virus, you can see here, it's taking over the cell's DNA, it's taking over the cell's proteins, making its own, ribosomes, make its own messenger RNA, making its own proteins. Over time, that cell will die, okay? Even though the production of or the presence of that virus is going to trigger the production of interferon, alpha and beta interferon, that doesn't really help this infected cell, but it will make the interferon and it will act on some neighboring cell that's not infected yet. And that neighboring cell will make these inactive antiviral proteins. And they'll just kind of sit there, ready just in case that cell eventually becomes infected. So like I said, even though this cell made the interferon, over time, it's going to be killed by that virus. When that virus, say it's a lytic virus, and it bursts out of that cell and now infects this other uninfected cell, it's got this inactive antiviral proteins, and what they do is bind to the messenger RNA and basically will stop protein synthesis for a time. Okay, so maybe for 3 or 4 days, that cell can survive without making any new proteins. And of course, if it's not making any new proteins, it's not making viral proteins either. So the idea is that this will slow the replication of viruses, and it will kind of give our other aspects of our immune system a chance to ramp up and target these virally infected cells. So that's not without some cost to the organism. Interferons cause some symptoms that we are all familiar with when we get a viral infection. Things like fever and malaise. That's a direct product of interferon. So it's not without some costs. Again, it's one aspect of fighting viral infections. Okay, and I'll just finish here with your book had a very detailed list of some other chemical defenses. I included it here just because sort of, hey, if you're interested in this, there's a lot of things that, again, we didn't go into this detail, but this might give you some perspective. You know, we didn't really talk about these antimicrobial proteins. We mentioned a few of these, like the lysozyme. We talked about complement and its function. We briefly mentioned a few cytokines here, a little bit more detail here. And these inflammation eliciting mediators, we're going to kind of briefly mention those soon when we talk about inflammation. Okay, so we will continue on Monday with talking about the different kinds of cells that function in innate immunity, and finish up with fever and inflammation, and then we'll be on to adaptive immunity. So thanks for listening.